Hello, everyone, and welcome to today's live broadcast, Choosing the Right Fit, Navigating the Options for NGS Target Enrichment, presented by Andrew Berry, Product Marketing Manager, Target Enrichment, New England Biolabs. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located on the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right of the presentation window and follow the process to obtain your credits. Please join me in welcoming Mr. Andrew Berry. I will now turn the presentation over to him.
Hi, thanks for joining today. My name is Andrew Berry. I'll be talking today about some options that we have in the marketplace today for target enrichment and, really how to navigate those options in terms of finding the right fit for the application that is needed. And because this is focused on human health, we'll be focusing mostly on that through this presentation. In terms of thinking about target enrichment and where it fits, I think it's important to start with an idea of where we stand today with regard to next generation sequencing. What you're seeing here is we, in the NGS, we see this type of figure a lot, and really what it's showing is the accumulation of sequencing data over time and how that's changed. And you can see the inflection point that happened sort of in the mid-2000s, 2005 to 2010, where next generation sequencing was developed and really started to dramatically accelerate the amount of data that's been produced. We've compared this in the past to Moore's Law, which really, is focused on doubling the data amount every 2 years. Next generation sequencing over the past 10 years or so has really outpaced that, and really to the point where we're doubling the amount of sequencing data every 7 months. And I think we joke about this sometimes, where we talk about the Moore's Law obligatory slide for NGS presentations, but I think it's important that we actually realize that we've actually sensitized ourselves to this quite a bit. In terms of really appreciating just how much data and how quickly we're changing the amount of data and accumulating these data over time. So the world's sequencing data is doubling every 7 months. And, I think just to put that in perspective, that means that, from today in April of 2018, by November of 2018, we are going to double that amount of data and have that available. And, the amount of insight that we're able to glean from this is really just scratching the surface. And I think that's an important thing to remember as we think about, the application of really looking at specific subsets of genomic targets in greater detail and really trying to understand that. So, just to, to put that in perspective a little bit, the amount of data that we have is actually going to be double in 7 months that we have today. And thinking about the different conclusions and hypotheses that we can test using those data. So what are we trying to do with all of this? I think, this is sort of a bit of a joke, but, the understanding is that, we want to basically accumulate all of this information and start to deduce some conclusions about the relationship between those genomic signatures, those nuances in the genome that make up the sort of 0.1% of the differences between us, and take that to the point where we can actually understand the impact on human health. So, maybe we're not actually looking at the inability to find the cell phone off button in a theater, but, understanding more importantly, the underpinnings of disease as it relates to humans. So, what we've really seen is, a lot of controlled studies where we're at the point where we're filling up databases with information relating genotype to phenotype and trying to draw logical conclusions about, not only what is the genesis of disease, but how does disease progress? How do resistance mechanisms work when we're undergoing a treatment for a given disease, and how different individuals respond, not only based off of their inherited genomic makeup, but off of things that they've accumulated through the course of their lifetime as well. And I think the best analogy that I can give is we're at the point now where we're really in the process of building a library, and a universally accessible library that one can go to try to find find out the information they need to be able to cure this. And what a wonderful time to be alive when these data are becoming available and able to use these. And a lot of the work that's happened in generating all of this data is really filling up those bookshelves, right? And filling up those libraries with books that are useful information, that are well categorized information that people can search and be able to use effectively to really test their hypotheses and build new ones, hopefully. Just a little example as it relates to target enrichment. So one of the more famous databases of genomic information is something called the Exome Aggregation Consortium. This has since been updated to be something called NOMAD. But what this is it's a really trying to aggregate the world's whole exome data, which whole exome data does fall into the category of target enrichment. But we're profiling the entire protein coding regions of the human genome. So these tend to be somewhere between 30 and 50 million base pairs of coding region that we're able to select out of the genome and study in greater detail. Well, this aggregation of this is an incredibly powerful tool, because basically what it does is it allows people to understand variant frequencies across populations and across geographies. So if there's a rare disease being studied on one continent, they can actually look at the variants that seem to be causal for that and try to find out on a more population-wide basis the frequency of those variants and whether or not they're associated with any known cases of a similar disease or a similar phenotype. So this is just an example, and I did some back-of-the-napkin calculations on this just to understand, based on the performance of whole exome data. So for these 60,000 humans, when we run whole exome sequencing, we're typically getting somewhere between 60 to 80% of the bases actually mapping to those targeted regions, so those regions that we're intending to capture that map to the protein coding regions. So 60 to 80%, that means that 20 to 40% of those bases are actually thrown out and thrown into the bin. And I think it was just looking at the data that we don't use for those types of studies, there's a significant amount of data that's accumulated, and looking at that relative to our ability to screen something like BRCA1 and BRCA2, which have really well understood sort of clinical phenotypes that can be associated with that. The amount of data that are in those 60,000 exomes that are in the EXAC consortium database, if we just took the off-target reads from that, we'd have enough data to actually screen the entire U.S. female population for BRCA1 and BRCA2. So I think this sort of highlights the importance of having targeted enrichment solutions that really work well and really are able to zone in on the regions that we're particularly interested in. So what we'll spend much of the time today is really thinking about the different options that are available there and how to really choose the best fit for a given undertaking. When we think about translational research, this is actually the definition from Wikipedia. But it applies findings from basic research, basic science, to enhance human health and well-being. It's in a medical research context, it aims to translate findings and fundamental research into medical practice and meaningful health outcomes. And I think this fits perfectly with where things stand today regarding the amount of data that's available, but also, the application of it and the challenges that arise from going from, a basic research study where you may have, plenty of high quality material to actually applying this in a real world setting where, samples that you have may be highly degraded or in very low input, very low amounts that are actually able to be used for analysis. And I think also important is to understand that this is a cyclical process, and that, the outcome and the things that are learned as the outcome of one study are really used to generate the next hypothesis, and that, we're really standing on the shoulders of giants and taking all of this information and trying to progress our understanding. And as we solve one problem, it typically opens the door to 3 or 4 more that we want to address. And I like to think about this in some ways as a funnel, right, between the initial discovery of sort of that association of, variants and genotypes that are associated with the given phenotype, and then obviously this process for how that might actually translate into clinical practice eventually. And as we think about the amount of content that's typically used for that, that tends to diminish as well in terms of, really looking very broadly. And we see that across the different types of organizations that are involved in some of these activities, as well as, the applications that are being used for this. And then finally, the tools that are being used to actually generate those data. So, we think about the different types of organizations, and from a discovery standpoint, this tends to be academic research, large government funded projects that are doing things like population scale screening, where we're looking at lots of healthy individuals, lots of affected individuals, and really trying to do that as broadly as possible to not miss anything, and using tools like whole genome sequencing, RNA sequencing, and whole exome sequencing to fill up those databases. when we think about it from the translational side, it really moves into things like organizations like hospitals and pharmaceutical companies that are looking to develop new treatment plans and, test out these things through clinical trial processes and actually test those hypotheses to understand if, the treatments that are being developed that are targeted towards a given genotype are actually, impactful. And for those, seeing things like comprehensive panels that might include things that we know have a definite impact, but also some things that we want to learn something from, is kind of the trend that we see there. And then finally, as we move to the real clinical application of this, we're really talking about hospitals and diagnostic service labs, where we really aim to return information on a patient back to a physician. This is really where we want to narrow in the focus of the type of content that we want to include to just those things that are going to be clinically impactful. So that's where we tend to see that sort of funnel shape coming into play. In terms of utility, I think, really one of the primary things to understand is what am I looking for? And understanding the difference between a germline or a somatic variant, obviously very basic, but this is going to impact the amount of sequence coverage and the coverage depth that you're going to need to achieve with a given target enrichment application. So, whether or not we're looking at broad scale enrichment versus very focused enrichment, who's going to be included in the study? Is it the individual itself? Is it the proband plus the parents in a trio? For somatic variant calling, we're not talking about inherited variants. We're talking about things that are really happening through the course of one's lifetime. It's the question is, are you sampling a biopsy of a tumor itself, and just looking at that? Are you going to have access to a matched normal sample that you can use to really do some error correction and understand which variants are actually present in the germline and subtract those out from your somatic variant calls? And one of the strategies that's been used there is this idea of having a panel of normals. We would use something like one of these broad databases like EXAC to use and look at sort of what is the frequency across the population of variants that are found, and use that as a way to sort of correct for some of the noise that is inherently present in NGS data. From a germline perspective, if we're looking to understand, sort of the genetic basis of things, whole exome sequencing makes a lot of sense because of the ability to mask data. So, rather than a laboratory having to run multiple different panels to look across different subsets of genes, run the whole exome, you will get sufficient depth of coverage to be able to call most of the germline variants from that, and then, only return back in your report the things that are being asked for. So rather than running lots of different tests in a laboratory, you can run a single test and generate all sorts of information that might be relevant for a given hypothesis test. In terms of the types of variants, I think this is also an important consideration in terms of the different technologies that are available. So whether or not you're looking for single nucleotide variants, looking for insertions or deletions that might be present, also looking at copy number variations. So these are all things that are going to impact, which technology you use. For example, copy number variant detection is something that's relatively easy to do with whole genome sequencing and even whole exome sequencing. But because you need to understand sort of the baseline performance of these variants and the baseline performance in terms of correlating the number of reads present for a given sample to a given phenotype, understanding that variation is important. So whether or not a target enrichment panel has sort of markers present to look at the baseline across the genome and not just be looking at a focused event that might be present. So copy number variants, also known as copy number aberrations, and a focal amplification is a common type of CNV that's detected in many of these studies. So I think, thinking more about the different types of approaches, again, there's our Moore's Law slide shown a slightly different way. But, even though sequencing costs have continued to decrease for oncology applications, for things that are really looking at somatic variant detection, we have to go deeper in terms of the amount of coverage that's present. So this table that you see there kind of shows the differences between targeted sequencing, whole exome sequencing, and whole genome sequencing, both in terms of the size that's being actually enriched for. And so, for targeted sequencing, looking at everything from, 100 kilobases or 10 kilobases all the way up to, 10 megabases. For whole exome sequencing, typically about 32, although there's some designs that go up to about 100 megabases. And then obviously whole genome sequencing, sequencing the diploid genome, looking at 3 gigabases and looking at that at different coverage depths. So for targeted sequencing, because we want to do somatic variant detection, we're typically looking at coverage, greater than 500x. So actually getting 500 reads for every loci that's present in that enriched sample. Whole exome sequencing tends to get you about 150x, depending on what that is. And it's important to understand that, what exactly the deliverable is for a whole exome sequencing, because it will impact whether or not you'll be able to use that for somatic variant detection. Whole genome sequencing, sort of the standard, is 30x. And again, this is typically good enough to understand the germline variants that are present. But if we want to look at somatic variants, we're going to need to go deeper. And then costs associated with this. This is, continues to be a dynamic situation in terms of the cost of sequencing and the cost of all the sample prep reagents. But, in terms of all-in costs, targeted sequencing typically runs somewhere between, $200 to $500 per reaction. Whole exome sequencing can range from $300 up to about $1,200, depending on, again, the coverage depth that you need to go to and the sequencing platform that's being used. And where whole genome sequencing, we're able to do this, including, sort of all the data processing for about $1,500, and that continues to decrease with new sequencers as well. So this continues to be a very fluid situation. And fortunately, it seems like the cost of sequencing continues to decrease, but that also can create some challenges up front to be able to manage the sample volume for that. One of the things that I think comes up a lot is this question of allele fraction and what that means in terms of sensitivity. So, variant allele fraction is something that really is telling us the frequency that a variant is present, which, for well-characterized truth samples are, very well understood. But for a clinical sample, you're really trying to get an estimation of that frequency based off the data that's available to you, which is typically trying to correlate the number of observations that you have of a given variant relative to the total number of observations that you have for the wild type. But that can change based off of a lot of different factors. And I think it's important to recognize that it's not just as simple as the number of reads relating directly to variant allele frequency. So, for one thing, tumors are a heterogeneous mixture of both stromal, which is going to have your normal genome, as well as the tumor cells in complex genomes. And the variants are present at a wide range of frequencies. So if we're thinking about a driver mutation that we're trying to detect very early in the genesis of disease, from a sort of prognostic standpoint, those are present at very low allele fractions, whereas, passenger mutations that might be, very frequent might come at a 50% allele frequency, depending on what they are. And then I think further adding to this is the fact that variants are typically present in amplified regions. So if we're looking at copy number variants where we have multiple copies of a given loci, that needs to be factored into the equation as well. And this, really impacts the depth of sequencing that is required to be able to call this reproducibly. And we don't just like to see a variant once. I mean, typically most people like to see a variant present in at least 10 reads before they confidently call it a true positive. So, when we think about that calculation, it's really a percentage of the reads calling a variant from, the analytical side. So that means that, for example, if you have 100 reads and the copy number is equal to 1, if the variant is present in 50% of the reads, you'll call that a variant allele frequency of 0.5. If your copy number equals 2, that means that you have twice the opportunity to observe this. And if it's present in 50% of the reads, that just means that you only have a 25% variant allele frequency. So in effect, the higher the copy number equals the lower allele fraction. And in terms of understanding the depth of sequencing that is actually required to call a specific variant, I typically try to think of a simple formula where, if you want to get 10 reads per variant to be able to call it confidently, if you have a 1% allele fraction and you have 100 reads, it's basically going to be present in 1 out of every 100 reads. So, you basically need to be sequencing to about 1,000x to be able to call that reproducibly. And as we think about things like, cell-free DNA and those applications where we really want to push the variants in terms of looking at these driver mutations early on, those tend to be down into the 0.1% where you really need to get high depth of coverage in order to have enough times to actually see it, because, you'll only be seeing that variant in 0.1% or 1 in 1,000 reads. And if you want to get 10 reads, that means you're going to have to sequence to a depth of roughly 10,000x. I think further complicating this is the fact that, you have false positives that arise from, really errors that are happening in a biological sense, that have to do with things like storage. So a great example of that is FFPE samples. So these tend to be highly variable in terms of the quality. We've seen samples from FFPE that look as good as fresh frozen tissue, that could be very old, but were collected properly in a clinical setting, were stored properly, that look very good. We've also seen recent FFPE samples that tend to look very poor. And again, this is highly dependent on the specific collection and the specific way that samples were processed. But there are, errors that are incurred through the process of actually fixing the tissue and actually getting it embedded in paraffin. And we see, it's one of the reasons that FFPE tissue is so stable is that, you are creating cross-links. So you see, lots of protein DNA cross-linking happening, DNA cross-links, as well as the actual damage where we'll have backbone breaks that'll include double-stranded breaks, single-stranded nicks, as well as, just gaps in terms of the content that's available. And then there's also sort of other impacts that will cause errors where you see things like hydrolysis and base deaminations that'll actually incur errors into those samples. Oxidation will happen where we'll actually get conversions of specific nucleotides. So in this case, from a deoxy G to a deoxy T, that are going to impact the sort of noise that's present in your sequencing data, and really, show, the types of, really make it hard to call variants accurately if you have that level of noise, and looking at the false positives versus true positives. And we'll talk a little bit more about some strategies that can be undertaken to overcome some of these a little later in the presentation. So, in effect, what this is going to do is those sort of top cross-linking and breaks are going to reduce the amount of material that's actually available for enrichment. And then obviously in actually incurring some of those errors. If we think about the actual process for going from FFPE, so coming from an FFP block where tissue has been stored and actually getting that onto a sequencer, typically that FFP block is taken into cross sections and sliced into sections. Typically, that'll undergo some sort of upfront qualification, be that through some, normal H&E histopathology, potentially immunohistochemistry using specific antibody labels, or FISH analysis to try to, actually look at what's present and do some actual biomarker discovery on that. So those sections are mounted onto slides and then going through this process. And then, if we actually want to get material off of that, somebody actually has to go in and scrape the actual tissue off of that, put that into a DNA extraction protocol, go through your target enrichment library prep process, and then finally get to your next generation sequencer. So it's not a trivial thing, and there's a lot of steps along the way where things can happen to the DNA as well, which can, again, diminish the amount of material that you have present for next generation sequencing analysis. Or cause damage that might induce some false positives into your data. Talking a little bit about cell-free DNA workflows, I think this is a very popular topic and very hot topic because of the potential of this. Obviously, being able to look at DNA that's being shed by a tumor, as it's, going through metastasis is it has some great advantages in terms of it's, much less invasive than a tissue biopsy, especially, depending on where the tumor is. Again, looking at this for sort of diagnostic prognostic applications, if you could have a blood test and tell you whether you have the potential to develop a cancer over time, the value of that information is huge. And there's a lot of things happening. There are some commercial tests available that have been, gone through sort of CLIA validation and are available to actually send your samples through and actually receive a clinical report. But a lot of this is still in the research side and really understanding the capability and the predictive nature of cell-free DNA. And a lot of these are looking at sort of longitudinal studies where they'll look at patients across the course of tumor progression, across, treatment with various therapies and trying to understand, what is happening in terms of the cells actually in the tumor actually developing resistance for a given therapeutic. So, understanding the nuances and the presence of cell-free DNA based off of those is something that is easy to do, again, because it's non-invasive. Important to understand that for cell-free DNA, it tends to be present at very low allele fractions. So, this is based off of the percentage of the cell-free DNA itself that is derived from tumor, and also the frequency of the variants that we're trying to detect being at low frequency. And that sort of 0.1% is kind of the target for most of these types of applications. The DNA itself is very fragmented, tends to come at a size of about 160 base pairs, which actually fits very well with next generation sequencing read length, and also doesn't require any fragmentation that can also lead to some sample loss. So, I think this is a really interesting area that will continue to be explored, and the sort of clinical application of this is only going to grow over time. And having technologies that are able to really push the conversion of molecules that are taken from blood blood and put that onto a library preparation or into target enrichment onto a sequencer to actually develop the depth of coverage that we need, and to do that as efficiently as possible because we want to get to such high sensitivity, low frequency variant allele fraction calling. So shifting gears a little bit to talk a little bit about some of the techniques that are actually used to actually run target enrichment. I like to break this down into sort of two main strategies, and the first being hybridization, the second being amplicon or multiplex PCR. And I like to separate them this way, and I think that there's a lot of variants of this that we'll talk about, but fundamentally, I think there's two separate strategies for actually enriching the targets that you're interested in from the ones that you aren't. And breaking it down between these two, I think, is an easy way to sort of bucket the two different types of technologies. So for hybridization, really what we're focused on for the enrichment is balancing the melting temperature of a long oligonucleotide probe or bait that's going to hybridize completely to a section of DNA. And what we use is a magnetic bead to pull that down. Whereas for amplicon sequencing, we're really relying on amplification of those targets to generate the enrichment itself. So rather than pulling things away and subtracting the stuff we're interested in, you're making copies of the DNA, which has some issues that we'll get into a little bit. In broad strokes, hybridization-based approaches tend to work better for larger enrichment panels up to whole exome and beyond that. They tend to have higher sample input requirements, and we'll talk a little bit about deduplication, which is an important factor when we're trying to predict allele frequencies and detect variants. For amplicon sequencing, this tends to use shorter primer pairs, so there is a melting temperature dependency, and because they're shorter, it tends to be more or less normalized across than it would be on a longer oligonucleotide as you're looking across different targets. It tends to have very short workflow time, so it's a very simple way of generating libraries based off of the fragments that you're interested in. Becomes challenging as you try to scale the technology, though, as you get to many targets and making sure that primer interactions aren't happening and different targets are amplifying evenly. That's where we can run into some issues there. But also tends to have very low sample input requirements and able to address things down to 1, 5, 10 nanograms input to a PCR, because you are using amplification as that primary mechanism of enrichment. So just to talk a little bit more about hybridization-based enrichment, this is originally published by Nurki in 2009, but in this instance they're using oligonucleotide baits. In this case, synthesized on a microarray. So these are synthesized. In this case, these are converted into biotinylated RNA fragments that are actually complementary to the DNA that we're trying to hybridize to. In parallel, there we start with genomic DNA that's been extracted. That tends to get sheared either mechanically or through some enzymatic fragmentation process. And basically what is done is there's a hybridization that takes place over anywhere from 18 to 72 hours, where those oligonucleotide baits are biotinylated and bound to the targets of interest, and then using a streptavidin bead are pulled away. Some PCR is performed again. Second round of PCR is performed to actually elute the enriched fragments off of the beads, and then that product is taken on into next generation sequencing. Important to note that a library is made in this process here, where you shear, you actually have to ligate your adapters on there, and then you typically PCR enrich that before you go into the hybridization. Mentioned it tends to be a longer process, which has some issues when you're trying to implement this in a more clinical setting, where turnaround times tend to be shorter, or there's a desire to have shorter turnaround times. And again, these work really well for large panels, down to about 5 megabases or so. Beyond there, what we start to lose is some specificity, where we see lots of reads that are mapping to our off-target regions will tend to come and come along for the ride and make their way into our sequencing data. So, as I mentioned, for a large panel, whole exome, somewhere between 60 to 80% on target is looking very good. When you start to get into more focused panels of down into the megabase or lower range, 30 to 40% on target is actually doing pretty well for this. So you're enriching for the regions that you're interested in. You're getting good fold enrichment, but there's a lot of other stuff that come along for the ride. Multiplex PCR is sort of the opposite of that. Again, it's a very simple process that uses primers that are specific for the regions of interest to actually amplify up those regions. There's a couple different approaches to this, and then people have, taken this and done lots of interesting things with it. But, primarily what this involves is having a forward and reverse primer, either having those primers themselves tailed with the next generation sequencing adapter sequence to make that library. So obviously a very simple process, or a PCR process where, PCR takes place and then you actually go into a ligation where you ligate those universal adapters onto the proximal ends of each of those molecules. I'd say the biggest challenge, even though it works very fast and uses low input, the biggest challenge is, primer interactions, having uneven coverage across the different amplicons, and, getting tiling across some regions that may be difficult to design a primer to. PCR duplication is also a challenge. I'll talk a little bit more about that, but, effectively what you have based on the primer sequence is a unique start and stop site. So it becomes very difficult to differentiate between a molecule that is a true biological replicate and something that is actually just a PCR copy that is going to, again, impact our ability to predict variant allele frequencies and understand, the prevalence of a mutation in a given sample. There's been lots of alternative strategies that have been developed as well. So selector probes are one, and this is where we have a probe that's specific to a region of interest. There's a circularization and a amplification event that takes place after that. Molecular inversion probes are a useful tool for this as well. Won't get into too much of the specifics of these, and then multiplex extension ligation is another example where people have adapted a amplicon-like strategy to, try to produce better quality data. And we see these prevalent in the marketplace. So as a product marketing manager, I am responsible for a product that fits into this space. So this is NEB Next Direct. This is a hybridization-based approach. So it's a little bit different in the sense that typically you make a DNA library and then do your enrichment. In this case, we enrich directly off of the genomic DNA. So we do the enrichment first and then take that enriched material and convert that into a next generation sequencing library through a series of enzymatic modifications. It's a single day workflow. I think, some of the benefits is that we incorporate an enzymatic removal of off-target to enhance the specificity for focus panels, and also incorporate something called the UMI, or a unique molecule index, that we'll talk about a little bit more, that is helpful for understanding the difference between a biological replicate molecule and a PCR replicate molecule. So when we think about the different metrics for target enrichment, I think it's important to understand what each of these sort of mean. And, sensitivity, and just as a sort of disclaimer, we're talking about, really the key metrics as an output of next generation sequencing, not necessarily related to any sort of an analytical specificity or sensitivity. But, when we talk about sensitivity, we're really saying, I like to use the analogy of a needle in a haystack. So from a sensitivity standpoint, what is the lowest variant allele frequency I can detect in a data set? And, a way to think about that is, what is the smallest needle that I can find in a big haystack? And this is, determined based off of multiple factors, including false positive variants and presence of those, the actual allele fraction and trying to get to very low allele fraction variants, as well as, we talked about some of the stromal contamination that can make it difficult to call variants based off of the actual cells that are used as an input into the assay. Specificity is obviously important, and I think that has to do with the efficiency of the enrichment. So, how efficiently can I separate my targets from the rest of the genome, or how efficiently can I separate my needles from the haystack? And you can see that image there where we have a magnet pulling the needle away. Then not coincidental. That's how we actually perform target enrichment, using magnetic beads in many cases. Uniformity is really important. So, how many targets can I find with equivalent efficiency in a data set? This is important because it ultimately dictates how much sequencing is required. So the presence of PCR duplicates will obviously impact that, as you need to, sequence past those duplicate molecules to get to the true biological molecules. Any PCR bias where there's different efficiencies in terms of the enrichment is going to throw that off, and can be difficult for balancing baits, specific baits in a given panel. Again, I think at the end of the day, it comes down to, weighing our options and saying, do I need a fast workflow? Do I need something that's less expensive, that works for low input DNA, has good specificity for my targets, but, is going to be difficult to scale content, if I need to add genes to my panel, if I need to look at a larger subset of genes, I may be dealing with poor uniformity across targets and overall a lower quality of data. Whereas with hybridization-based enrichment, you tend to have higher data quality and works really well for large panels, but it tends to be a longer workflow. It's more expensive, tends to require more input DNA, and as you get to smaller panels, can have low specificity. When we think about coverage uniformity, this was actually just published from a publication that came out last month. This is looking at comparing hybridization-based enrichment to amplicon sequencing. So what we're looking at in the figures on the right is actually the uniformity of coverage. So the mean target coverage for the same panel was designed both with capture probes as well as amplicon enrichment. And, what we would love to have is as normalized as possible, where we're getting, even coverage that's equivalent to a given value. So we'd love to see the straight horizontal line in terms of the coverage. But what we see here is for capture probes, for hybridization-based enrichment, you tend to have more even enrichment. Again, because we're selecting those targets away from the rest of the genome as opposed to amplifying the targets that we're interested in away from the genome. And you can see the coverage variability. So even in the scale here, we see the coverage in terms of the standard deviation about 212x, where, when we get into amplicon sequencing, you're talking about thousands of fold difference between those. And, that is going to have an impact on what you actually are able to do in terms of trusting the variant allele frequencies you're trying to predict, the fact that it's going to require over sequencing. So for your high performing amplicons, you're going to end up sequencing those to really high sequencing depths if you have uneven coverage to get your lower performing targets up to the minimum threshold where you can reliably call variance over frequency that's needed. So it's just an important factor to understand as you're thinking about the strategy to choose. PCR duplication. So, what you see in that figure on the left, those are integrative genomics viewer plots looking at coverage profiles for something that was done with hybridization-based enrichment versus amplicon. And again, one of the benefits to hybridization-based enrichment is that you tend to have non-uniform start and end sites because we're randomly fragmenting DNA. We're hybridizing a probe that may have a certain amount of homology to that actual molecule, using that to pull that away from the rest of the genome. You're going to end up with random start and stop sites for each of the reads that you're actually generating out of your sequencing data. Whereas for amplicon, because it's based off of primer design and primer sequences, those amplicon reads are going to have the exact same start and stop sites. Again, that makes it difficult to differentiate between a PCR duplicate and an actual biological duplicate molecule. On the right, I think there's an interesting plot there is the way that we actually can look at coverage and having non-uniform start and end sites are important because it really allows us to determine the true coverage that's present. And when we look at this, so in this plot, what we see is any given metric, but typically what we would look at is unique coverage that would be present for a given loci. And really, what we want to be able to do is increase the amount of sequencing coverage, the amount of reads that we have, and continue to add new information into that. So in that purple figure, you can see that we can continue to add sequencing depth to that and actually find some new things. Whereas in that green line there, basically what you have is you've exhausted the molecules that you have for next generation sequencing. So you can keep sequencing, but all you're going to be sequencing is PCR duplicates. So you can continue to add depth, but you're not really adding that. And the challenge there is that with amplicon sequencing, typically you don't know because you really can't understand what the true duplication that's happening for a given data set would be. So you really don't know whether or not you can continue to sequence all you want and find high depths of coverage, but you may not discover any new variants that might be present in those samples. One of the strategies that's been employed to overcome this is this idea of having a unique molecule identifier, a unique molecule index. And that's, again, what that's going to do is mark the molecules with a DNA barcode before you amplify so that you can understand the genesis of each of your resulting molecules and look back at that to be able to understand the duplicates that are present. In addition to that, there's some strategies that are employed that actually utilize unique molecule indexes to do error correction in something called consensus variant calling. And just quickly, I think, understanding how this works. So without the UMI present, basically, what we have is two biological replicates of a molecule, copy A and copy B. We're going to perform PCR on both of those. So what you see in the red is a true positive variant. It's going to get perpetuated through that. But in that process of doing PCR amplification, you're also going to acquire some false positive variants as well. And, in this situation, because we don't know which of these are which, and those molecules effectively look the same, it's really difficult to understand which of these variants are derived from false positives, or which of these are actually true positive variants that we actually would want to include in a data set. If we take the same situation, but now we've actually tagged each of these molecules before PCR amplification, again, we're going to go through the same process of amplifying those fragments. We're still going to acquire our false positive variants that will come into play there. And what we have now is a situation where, what you see is we're able to group those reads by the actual UMI. So that would be a sequence that would be added to the read itself that would be, allow us to group these things and call what we call read families and look across those to say, okay, in this situation, I can see that for the UMI, the yellow UMI, basically all of my DNA molecules are harboring this red true positive SNP. Whereas in the situation where with that sort of teal UMI, basically the variants that are not present and tend to be variable across that. And we can identify those ones that are variable as being high false positive. So at the end of the day, what this is going to do is reduce our signal to noise ratio when we're actually trying to call true positive SNPs. It's going to lead to higher sensitivity variant calling and more accurate assessment of variant allele frequencies. And one of the questions that's come up because, PCR duplicates have always been, viewed as a somewhat negative result that will happen from doing PCR amplification. We're now actually having multiple copies of these molecules is present because is helpful because you can actually use them to derive some useful information out of that. This is a data set that was produced internally here, but basically looking at the number of reads in terms of the consensus depth. So how many of each read, each sequencing read do we keep? So you can see that on the x-axis, as we go up from 1 to 3, you can see a dramatic impact in terms of the error rate or the Phred score. This is on the Phred score scale, but dramatic impact on the error rate that's present in terms of the quality of those reads. So we're able to get up to a Phred 50, very quickly where, we're down into the sort of Phred 30 range. As we do that. So that means that, again, PCR duplicates or duplicates in general are a friend as long as we can identify them as PCR duplicates and use them in conjunction with UMIs to be able to do this type of consensus variant calling or error correction. And I do think this is, one of the things that will ultimately help target enrichment strategies, really take it to the next level and be able to look at really low allele fraction variants that might be present in some of these samples. Again, as we push from diagnostic into prognostic type applications, being able to really push the depth of sequencing, push the conversion of those molecules, so we can actually make sure that our reads are getting to, the data sets themselves are rich enough that we can actually derive some of this really useful information from. So I think, just in conclusion, there's really a lot of considerations that need to be taken into account as you're deciding, what the right fit is in terms of a technology for a given application. Understanding the goal of the study. Are we looking for somatic variants? Are we looking for germline variants? Do we need to call copy number variants with this? Sensitivity is huge because it's a direct function of how much sequence depth you need to get out of your actual sample and, understanding the percentage of variant allele frequencies that you need to get down to in terms of sensitivity. Input material, as we discussed, can be highly variable, but understanding not just the type of material, but also the amount of material that's going to be available for your study. And also the panel itself. How big is the panel? How many genes do you want to include? Are there intronic regions that can have interest, different challenges that might be associated with homology to repetitive elements in the genome versus, looking for just the coding regions in those exonic regions? How many samples? How scalable is my technology? Can I run this across, thousands of patient samples? Or, is this something that I really just want to run a handful of things on? What is the turnaround time for the workflow? How fast do I need to be able to return data out of this workflow? And how easily can I automate this? How easily can I scale this technology if I do need to implement it into routine practice? So again, it's there's no single technology, I think, that really covers the gamut of what people need to accomplish with target enrichment. But it's clear that additional development does need to continue to take place, and this is an active area where continuing to innovate and continuing to push the boundaries of what's possible is really important to be able to recognize some of the benefits from all this wonderful genomic genomic data that's being developed and generated through many organizations around the world. So with that, I'll thank you, and I think we'll move on to the Q&A section of the webinar.
Thank you, Mr. Berry, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what applications are there for target enrichment outside of translational research?
That's a great question. I think, it is obviously focused, this presentation was on sort of human health and how that applies there. But target enrichment is being employed across industries and everything from, plant genotyping, crop development, things like food testing, understanding, different breeding strategies for different organisms and cattle and things like that. So there's lots of different applications for which target enrichment is being applied. And some of these will have different requirements based off of the needs of a specific application. But I think it's as much as the imagination can take it in terms of fundamentally needing to look at the genome of an organism and look at regions of that in greater detail than you would by just doing whole genome sequencing.
Great. Our second question says, how many base pairs long should a UMI be?
So yeah, the question about how long a unique molecule index, really this is going to have to do with the size, so the operating size of the UMI. So if we think about it very simply, it's a random sequence that gets appended to each of those molecules. So how long that is going to impact the number of available UMIs that are there. Some studies have shown that having a UMI greater than about 8 base pairs is useful. I know in our technology, NEB Next Direct, it's a 12 base pair UMI. So that ends up being 4 to the 12th, which is just under 17 million available. But most of the things that I've read really recommend having a UMI that's about greater than 8 base pairs long.
Okay, our next question says, how much DNA can you get from FFPE?
Yeah, so FFPE tissue that we talked about a bunch is a pretty common sample type for clinical workflows. Obviously it allows things to be preserved. The actual amount of DNA that you're going to get out of that can be highly variable, based off of obviously the biopsy itself, and the amount of tissue that was embedded is going to impact that. But also, how it's being utilized, whether or not you're taking a section, how big that section is, if you're doing a punch where you're actually looking longitudinally through that section, that is going to impact that. So, it can be anywhere from a few nanograms, 10 nanograms of input to several hundred nanograms if you actually have a whole block available. So it really does, vary quite widely, but somewhere between sort of 10 and 100 nanograms is typically what we see for DNA input coming out of FFPE tissue.
We have time for one more question, and this one says, what are the common sources of error in target enrichment data? It can come from different places. Obviously, we talked about some of the errors that can be incurred during the sample processing itself. Through fixation processes or extraction processes where potentially treated with harmful chemicals that can actually induce some base pair changes to the actual DNA, as well as impact that backbone itself. Also, PCR amplification is another source of error where as you're doing PCR and making copies of things, it's not always perfect. So you're going to incur some level of errors that'll happen there. Also through many of the hybridization-based enrichments, when we're doing the washes to remove off target, there tend to be some, relatively harsh chemicals that can be used there that can also induce some base changes and cause error in your sequencing data. And then finally, on the sequencers themselves, right? So sequencers have inherent error rates, and some of that comes from the sequencing process itself. Some of that comes from a clonal amplification that'll take place before you actually get onto your sequencers. So, all along the way, we're doing things, and as much as we can do to actually preserve the integrity of that starting DNA sample by not subjecting it to multiple rounds of PCR. Hopefully, getting things from clinical collection as quickly as possible is only going to help reduce those errors that might be present in NGS data. I would like to once again thank Mr. Berry for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through July 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.